So based on the size, of course, this is a little bit more informal. The tour was great. Did we have any questions moving forward before I dig in? Anybody have any questions? No? Okay. All right. So I'm going to really diving into systems biology. I am going to, based on time, of course, kind of present some broad strokes. As we've already kind of indicated this morning, our mission is to really leverage cutting-edge technologies in the precision health space. And fundamental to this, of course, is systems biology, and we'll talk about all the components that we use for that. Particularly, my expertise is in multiomics. So before we kind of dive into this, of course, we have to know what systems biology is and how it pertains to precision medicine. Sure.
Only because I didn't know this until a couple of months ago. Could you define what omics means?
We'll get there. Okay. Yeah. Actually, no, that's a great question, and yeah, that's yeah, we'll definitely get there. Okay. So systems biology is an interdisciplinary approach or a disciplinary field that aims to understand complex biological systems by examining their interactions. And that's really, really, really important when we talk about omics. And as I said, we'll kind of get into that. So of course, we know how this was really kicked off, at least in public awareness, with the Human Genome Project. So kind of leading up to the early 2000s, the 90s, this effort was spearheaded by several different organizations, and we were able to successfully profile the entire human genome. And actually, we see a hard copy printed out here. I mean, obviously, we're not doing that for any type of a data analysis, but it kind of gives you an understanding of just how much data we're dealing with. Computationally, we're talking about between 700 megabytes to a gigabyte. Now, when we go into my field, like the microbiome, and we're able to do really, really deep sequencing. So not only are we just looking at one genome, we're looking at the metagenome. So that comprises all of the microbes that we're able to detect and analyze. We're talking about hundreds of gigabytes or even terabytes of data. So obviously, all this data, while it can be very, very rich, we have a very complex system that we're working with. So in systems biology, as I said, it's interdisciplinary. We're really looking to kind of bridge everything together. I think a good analogy is kind of like a car, right? You can imagine trying to understand how a car works by only looking at one gear. So we might look at one layer within the multiomics paradigm, like the genome, for instance. But you're only really grasping its function. You're kind of missing the bigger picture with all the other interactions that are occurring with other components that actually make the car move. So systems biology takes a similar holistic approach. So getting at your question, what is it when we talk about omics? So in systems biology, this is kind of known as the dogma, the central dogma of systems biology. Everything is kind of downstream from the genome. So I am using a picture here from my work with the microbiome. So again, we're looking not just the genome, we're looking at the metagenome. So highlighted is that annotation for each one of those layers. So obviously we have the genome comprised of genes. That's what we refer to as genomics. We have the transcriptome. We're talking about RNA. Okay, that's transcriptomics. We have the proteome, talking about proteins, proteome. And then we have the metabolome, the metabolites, and that comprises the metabolome. So in terms of what we're looking at with how a lot of medicine is currently functioning, it's, I'm not trying to dismiss clinical blood work. It's definitely fundamental to how physicians are running their practice, and we're not trying to replace that, we're trying to augment it. But when we look at different blood chemistries, of course, this is kind of a reductionist approach. We're very much limited in terms of what the current assays are able to capture. And of course, you have the addition of, and again, not to take away from the physician, but they have to interpret everything based on their clinical knowledge to really understand what is going to be the best treatment option for their patient. Now, as we get into the omic age of the last several decades, not only do we have all this new data, which has really kind of opened up Pandora's box, and I think this, in many ways, has made things challenging and very complex for physicians, but we also have, of course, an increase in all types of different diseases, not just because of what we're having at a population level with just health going south in general, but also we're able to detect a lot more now with these omic type analyses. So all this together is really increasing physician burden. And I know in this space, genetic testing is very popular. Other testing is being onboarded. It's really trying to find out how can a physician actually properly leverage this tool or these tools to do something with it, right? So again, going back to the central dogma of systems biology, everything is downstream from the genome. You know, I never really got the term dogma. I don't know why we have to prescribe some kind of religious connotation.
Or be dogmatic about it.
Or be dogmatic about it. This is just kind of the way things are, right? So systems biology, of course, has applications in various areas of biological research, including drug discovery, personalized medicine, and understanding complex diseases. And really, I think the utility of this boils down to its shift from these reductionist type approaches, like clinical chemistries, for instance, and really trying to understand what's going on in terms of phenotypic expression. So this is a figure I think Panisse mentioned on our lab tour. These are all the known metabolic pathways in Homo sapiens. So this is actually from the Kyoto Encyclopedia of Genes and Genomes, KEGG, the egg. You see, it's kind of, we always have to, in bioinformatics, you always have to have funny little acronyms and symbols and stuff. You'll see a lot of them for the next couple days. But anyway, we see that things are extremely complex and, of course, interconnected, right? You might look at one particular pathway, you might look at a series of genes, or you might look at a series of metabolites, but all these different pathways are really showing us just how reliant these pathways are on basically talking to each other. So this presents a lot of challenges, particularly on the bioinformatics side, when we have several different elements when we're trying to actually condense this into something that, again, going back to the health modules, making something actionable for the physician. The integration, which is very, very challenging when we're integrating genomic data, transcriptomic data, proteomic data, metabolomic data. Then we have to do, once the data is all harmonized, collated, and conditioned, we have to do multi-scale analysis. So I'll refer to this really as multi-omics, because multi-omics is the idea of bringing together these different layers and conducting these analyses. And for that, based on recent advances in computational abilities, we're able to really do this type of analysis basically because of AI and machine learning. Machine learning, I would kind of classify for our purposes as a subset of AI. And we actually use a lot of network-based approaches for that. And this system has incredible predictive power, particularly when we're able to take longitudinal samples. Going back to what we normally do with clinical blood work, of course, we can take longitudinal samples with a patient, but you're not really using that data beyond your own clinical interpretation. The power of these AI systems is to get more precise for each time, each sampling collection, because we're able to run our simulations. So the power of our testing really comes in with these iterative cycles that we're able to perform. So really, it's taking all that complex data, funneling it down, and giving you markers, biomarkers, right? And those can be informed at the health module level, like we have in our report. They can be informed by clusters. I think Panisse is going to be presenting some factor analyses, which are really, really cool. And I'm excited to see some of your work there. But multi-omics, actually, I should have first asked this question. How many have heard of multi-omics? Okay. Oh, Gail, you're okay. So multi-omics really is the realization of systems biology, in our opinion. So if we look at what's been published in the National Library of Medicine over about the last 20 years, we see a massive increase in all the different layers. Of course, genomics has been around for quite a while, transcriptomics, et cetera. Even the microbiome has really exploded in the last 10 years. But if we kind of hone in, there's this little sneaky black horse coming up that has exponentially grown over the last 10 years, and this is multiomics. And everyone is trying to struggle in this space to understand how they can use multiomics to inform this research. And going back to what we kind of mentioned at the forefront of today, you know, I'll tell you, you know, I still have one foot in academia. It's very frustrating. I think I heard some conversations about the currency, right, of an academic is papers. We're publish or die, publish or die. That's fine. Oftentimes, a lot of what you get in peer-reviewed journals is basically garbage. But that's kind of the mantra of academics. And what we really wanted to do is not try to just publish, publish, publish. While we do that, I think we do it pretty well. We're trying to actually build usable tools. And to do that, we had to step outside of academia and really build this, basically a parallel institution. So while we're utilizing those resources, we feel that we can't do that in academic space. What they want is what we want, but it's going to take them decades, whereas we feel if we're more agile, we're connected with the right partners, we can do this a lot faster. And I think a lot of people are starting to catch on and realize this. So kind of looking at each level, of course, I think everyone in this room is familiar with the genome. This unveils specific genetic variations or mutations influencing genetic predispositions, inherited traits, susceptibility to diseases, and medication response. And of course, we know that genomic differences are a major source of variability in drug pharmacokinetics and response. And this individuality, this interperson variability, of course, is very important for what we are doing. So just as a quick example, of course, we know that there are you know, several different genes that we can look at, like cytochrome P450, that constitute a major enzyme family capable of catalyzing the oxidative biotransformation of most drugs, as well as other lipophilic xenobiotics. And we have a wide swath of how people actually respond to a lot of these different drugs that work off of this. We can have poor metabolizers, we can have extensive metabolizers, we can have intermediate metabolizers, and even ultra-rapid metabolizers. Now, while this is really important to look at, we have to remember that a lot of medications are taking an oral route. So while genes are important, these drugs oftentimes are coming into contact with many of the microbes that are inhabiting our gastrointestinal tract. Now, granted, most of the biomass of the microbiota is located in the colon, but there are microbes that have significant influence more proximal to the mouth, basically, in the alimentary canal. Of course, they have to cross over the epithelial barrier, so they're coming into contact with those cells. They're going into circulation. There's metabolism occurring at the liver, and then, of course, where they're going in terms of the target of the drug. So there's a lot of interaction going on, not only with the host, obviously, but with the microbiome. So while we look at genes, we're really missing the bigger picture in this overall systems biology framework. Now, the omics man, as Panis said, this is kind of a running joke with us. So you will see the omics man a lot. We maybe should have done, you know, one of the Truvian man. We'll work on iterations. But basically, really important for what we're doing and in terms of profiling is understanding longitudinal expression. So by and large, the genome is pretty stagnant. Now, it does serve as a fundamental starting point in omics because it's kind of anchoring the flow of information, helping us kind of pinpoint origins of pathologies, whether you know, genetic, environmental, developmental, et cetera. But really, the integration is the powerful part. So if we look at, for instance, the transcriptome, we know that this is actually quite a sensitive level when we look at factors that are sensitive to treatment, environment, and different health behaviors, often necessitating a lot more regular assessment. This is a pretty interesting study published in the Proceedings of the National Academy of Sciences back in 2018, looking at the difference at the transcriptional level and those that were, kind of living a normal life in terms of circadian rhythm and night shift workers. And they found about 3% of the transcriptome was shifted up and down, which, that may not sound like a lot, but in terms of functionality, that is a tremendous amount. So, and this was occurring in just a couple days. So in terms of sampling, of course, it's going to vary by case what you're specifically looking at. But this kind of really lends itself to the idea of when to sample and how often to sample. So proteomics, of course, we're looking at the proteins comprehensively, including their expression levels, their post-translational modifications, interactions, and functions within a biological system. Again, this is really important when we talk about function. PTMs, of course, that we all know: phosphorylation, glycosylation, ubiquination, acetylation, methylation. How many people have used some form of methylation testing? Have you found it to be pretty good in terms of informing you? Have you? Okay. Yeah, absolutely. So that's just one part of PTMs, right? So I think this is really kind of showing us the power of incorporating all of this data. Again, kind of going back to the KEGG database of all the known pathways, that's obviously a lot. As Panisse alluded to, right now we are covering 68% with our current offering of the Aristotle test, which measures 126 sentinel metabolites. So as you can see, it's pretty broad coverage in terms of what we can actually map out. Now, comparing that to what you would normally get with a pretty comprehensive clinical blood workup, looking at complete blood count, basic metabolic panel, your lipid panel, liver functional test, and thyroid functional test. While you have decent coverage, it really kind of pairs in comparison to what we're able to capture with the Aristotle. Now, another test that I know is pretty popular in this state or in this field is the organic acid test. Is that another test that you've used?
I haven't done that one as much as I've done the methylation panel.
Okay. So again, that even adding that, so now we have 6 different tests. Again, we're not really able to outcompete what we're able to capture with the Aristotle test. And again, you're getting all these separate. So the burden is on the physician to interpret all of this data, where what we do with our bioinformatic pipeline is actually make this into usable information with our health modules. And I'm actually going to show you some pretty exciting things that we have in the works and actually interrogating this data at a much deeper level with our conversational health agent. I'll do that towards the end.
Got a question.
Yes.
How much of the map do you guys think you're going to be able to cover in the next couple of years, let's say, with Aristotle?
Yeah, the V2 we're really excited about because we were just showing you the GCMS, but if we get the LCMS on board, liquid chromatography, we're looking at around 88%. So we're not claiming that we're going to be able to cover everything, and we're not trying to replace those other tests. We're just simply showing you our abilities right now. And this is all very iterative. We're working on in many different domains right now, all exciting, but we see a very bright future. We're biased, obviously, but we see a very bright future in the next couple of years. So shifting gears a little bit to my area of expertise, the microbiome. So how many are familiar with the microbiome? Have you used any type of testing? What type of testing are you using?
So when it comes to like home delivery kits and that kind of stuff,
Mhm.
I use Flore.
Flore, yeah. They're actually based out of basically ASU, right, with Rosa.
They're an ASU-based company. Yeah.
How's your experience been with them?
Pretty good.
Pretty good.
Pretty good. Yeah.
Yeah, this is a lot more competitive of a competitive space in comparison to metabolomics. But I think the power is really bringing together, as I was saying, with our multi-omic analysis. Whoop, went a little bit too quick there. So if we actually look at the gut microbiota, it's quite small in comparison to actual biomass, but it's much larger than our own. Its metagenome is much larger than our actual genome. It's actually about 200 grams. If we took out all the microbes, you could actually kind of fit it in the cup of your hands. However, it's estimated to affect in some way approximately all the metabolites that you're going to find in circulation in your body. And this, of course, is kind of intuitive. It makes sense, right? Considering the locale of all these microbes, as I was saying, populating the alimentary canal. So these microbes are tightly intertwined with our health. They have a host of functions. We're really focusing in on its interaction with nutrition, whether it be digestion or absorption, and of course, inflammation. We know that approximately 70% of the immune system is actually located around the gastrointestinal tract with GI-associated lymphatic tissue. So there is a lot to look at here. We're going to have some overlap, of course, with the Aristotle, but this is really giving us a good idea of how someone is obviously interacting with their food based on all the metadata that we're able to capture with our surveys, and then what's kind of going on from the gut into the body. We know that not just the epithelial barrier, which everyone really kind of focuses it on in terms of disrupting tight junction proteins and having things like endotoxin enter circulation and spur inflammation, but also very important is the mucosa, the two layers of mucus that are actually kind of pushing the microbes away from the epithelial cells and forming a protective barrier. And there's actually specialized microbes that we'll go into a little bit later with a case study that are very important and almost act as stewards for increasing the health of the mucus. So being that we're still affiliated with ASU, we've done a lot of research in college students here. And this is a unique population because you often think of college students as adults. I guess you can. We looked at freshmen here in this particular study, actually just right over there down the street in our dormitories. So they're in a decently controlled setting. This was observational. However, they're still technically adolescents. They're still developing. Their microbiome is still kind of formalizing, not to the extent that you would get with, say, a newborn. But profiling their microbiome over the academic year, we noted tremendous variability in the profile of what type of microbes were inhabiting their gastrointestinal tract. In fact, we had 4 distinct branches, primarily Bacteroides. So I don't really like the term dysbiosis. I like to kind of look at the phenotype. So I'll say more like maybe disease-associated or health-associated microbiome. But basically, the core of the group was showing high prevalence of Bacteroides, which is generally associated with things like higher BMI, lower fiber intake, things that we would generally associate with disease-associated exposure. We also had a group of individuals that were quite healthy, had low body fat, were in the normal BMI range, that had a very significant and distinct branching when we look at the overall community. And these individuals harbored a specialized microbe called Prevotella, which is generally associated with health. So bridging this data with metabolomics, we were actually we were able to start looking at some of these multi-omic type analyses, and we noted that those that were clustering in the Bacteroides associated, what we call enterotype or microbiome type, had elevations in things like cholesterol, p-cressyl sulfate, even cadaverine. So these individuals were really showing perturbated metabolism coming from their gut microbiota. So going back to this particular figure, which is actually now, this particular publication just got accepted, so it will be in press pretty shortly. And actually, that paper in Biomedicines, we can send it to you all once it is in press. And usually Biomedicines is pretty quick. I feel like next week it should. Yeah, next week it should be up, so we'll send it to all of you. I mean, it really goes over kind of the meat and potatoes of this particular talk. While it's a little bit more hypothesis-driven, it's where we foresee things we perceive things moving. Now, there's a lot of challenges when we talk about integration. So you can gather genes, you can gather the genome, data from the genome, you can gather the proteome. All those different data types aren't necessarily not just in a similar format, but amenable to being collated and harmonized. So there's a lot of back-end work that has to happen before we can even get to the selection extraction phase, step 2 there. Once that is done, number 2 is actually really important for us anyway, because here we're actually looking at things that matter. Okay, so we might see elevations or perturbations like we just saw on the last slide with particular microbes. Of course, their function, which is really, really important. I think in many of these fields, particularly in the microbiome, we've been kind of bottlenecked for many years because we're just simply looking at microbes and inferring what they're actually doing in the body. It's really the function that is important. What the heck are they doing? Not just in the microbiome, but to our own physiology. And looking at things like fecal metabolomics, which we will be offering in our microbiome test, which is a distinction from other tests out on the market. So the metabolites that they're biotransforming, modifying, or actually even creating themselves, which are very important, particularly when we look at factors of nutrition. Most polyphenolic biotransformation is actually occurring in the colon from microbes. We're developing vitamins like vitamin K from the microbiome. So it's very, very important. And then, of course, what we're detecting in circulation with the blood metabolome. So it's understanding those interactions where we can actually have selection and then extraction. And from there, we can enter those features into our models to condense them into these digestible health metrics. So has anyone done, I know, Gail, don't raise your hand. You're kind of a special case. So has anyone gotten their results back? I know you just took your test. Did you do the Aristotle? Have you done the Aristotle yet? Okay, I'll say that for a second then. So we can get these different health metrics. So currently in the Aristotle, we have 12. I think the value for physicians is really going to be in our platform that I'm going to show towards the end, where you can get, of course, the health metrics, but you can interrogate each health metric, and of course the overall data to answer your specific questions to inform your care. So this study, yes. So this is another study that we did here at ASU, again with college students. We use the resources that are available here. We looked at a very important factor that not many people are studying, at least at the molecular level, which is screen time. So everyone in our modern era, of course, is exposed to screen time. It's extremely pervasive. We were very interested in looking at the effects from high screen time versus low screen time at the level of the microbiome and the metabolome. So of course, we can do all of our traditional analyses. We really like to do enrichment analyses looking at functionality. Again, moving beyond just annotation of particular features, but what the heck are they doing? What the heck are they doing, right? But that's just looking at each level independently. So for instance, here we were looking at the metabolome. We saw enrichment in these particular pathways. But how are they interacting? How is the microbiome interacting with these different metabolites? And for that, again, this is really kind of the challenge, and not many people have this figured out or a good system for this. We used neural networks. Okay, so neural networks are basically, it's a form of AI, subset in machine learning. We're actually able to form these nodes and perform thousands of simulations to see what basically is co-occurring, associating, and really pull out the strongest predictors in terms of what might be shifting the metabolism. And we noted 5 particular microbes that were really kind of pushing the community when we were looking at those with high screen time. And from this, we can actually form models. At the molecular level, the metabolic level, to understand what the heck is going on, moving beyond just annotation. And with these particular microbes, we found them to be elevated in individuals with high screen time, which was actually perturbating mitochondrial function. So here we see some real takeaways in this research. Actually, it was led by you, Penise, if you remember. Yeah, your favorite publication. Actually, we like these kind of little side projects, and we like to go into spaces that, no, I mean, it's intuitive to look at screen time, but no one's really going into these spaces.
And this was a project that was for a microbiome data science class, and they said, well, bonus points if you can publish it. And so we ended up publishing it, and what
In 6 months, I think we did everything.
we did everything,
Yeah.
we did everything. So how do you take, you know, this is really where Alex's skill comes in, is how do you take that middle figure and turn it into the figure on the right? Because that middle figure contains all the data you need to then map it to pathway.
Yeah, so this is really getting at feature selection, like I was showing you in that previous slide, and then taking this and actually mapping it to metabolism in a different health module. This slide. So once we get to the health metric phase, and I'll show you the Aristotle, this is where things can really be reshaped and then offer something meaningful to the customer, to the patient, or to the physician. And this is just kind of what we have offered in our current Aristotle test. So kind of our dashboard shows you the 12 health domains that we have, where you are in terms of what you could improve. We go from obviously, poor all the way up to optimized in terms of these health scores, spanning aging all the way down to mutational load. And then you see the metabolites that are informing each one of these modules. So when you look at like a traditional clinical blood test, you're looking at, obviously you have a value or a score, and that's plotted against the standard distribution of a population, right? So while that's valuable, the power of this really comes down to digital twinning and comparing the person to themselves. And we'll go over what we're talking about with digital twinning. So when you see this and you're making improvements, it's really based on what you can achieve, not some population norm. And I think this is really important when we talk about the variability that we get between people.
So excuse me, like on that test there, where you've got inflammatory, is that specifically related to inflammation in the gut?
Oh, this is the Aristotle. I'm sorry. This is the metabolome test. Yeah, sorry about that. Yeah, yeah. So of course there are a lot of challenges in this space, and as I was saying, recognizing individual variability is really going to be, I think, the game changer here. So a lot of tests currently, like the microbiome test from Flore, I believe they show you like abundance plots where you compare to different, like a population norm. Maybe it's set against like someone in your age group or whatever, right? So that's kind of missing the boat in our opinion. I mean, and we've done a lot of that type of research. This is actually a recent publication that we have from in Nature Communications where we looked at intermittent fasting, right? Again, we like to publish still, and we're aiming a little bit higher. So say what you will about these different journals, but we still got to play the game. So in this particular study, we compared two groups. This is a randomized controlled trial over 8 weeks, so it wasn't very long. However, we saw some really important shifts in not only the microbiome, but also the metabolism. So here we were looking at the microbiome and the plasma metabolome. And one group, the group that is colored there in, I guess it's like a magenta, was following a calorie-restricted diet, about minus 20% of their normal caloric intake, and on a kind of what the USDA would term healthy. It was basically like a Mediterranean type diet. The macronutrient ratios were all in range with what the USDA guidelines prescribe. The other group intermittent fasted 2 days per week. It was a modified intermittent fast where they were reducing their total caloric load by 75% on those 2 days. So it wasn't a water fast. Like I think you're on a water fast right now. Yeah. Well, this is similar. I'm sure you're pumping ketones right now, so you can hang with me on this one. But basically, we found performing a multi-omic factor analysis, which basically works in a Bayesian framework, 8 really important latent factors. We can't really see here too well, but basically, in terms of explained variance, we found that the plasma metabolome was close to 40%, showing variance between these two groups. So this is just plotted out in a PCA, whereas the microbiome only accounted for about 18%. So actually, in terms of the changes that were occurring, a lot of it was in the body, in the metabolome. And two important features emerged, factor 1 as a latent variable and factor 6. And factor 6, when we look at this plotted against different features or different factors with our participants, was negatively associated with body weight, BMI, waist circumference, body fat, really importantly, visceral fat, as well as other things like total caloric intake, carbohydrate, and sugar intake. So, it kind of is intuitive when we look at a lot of the fasting literature and what we see occurring metabolically. But when we actually look at these factors and what could be driving some of these changes, we saw a lot of changes associated with intermittent fasting and factor 6 related to metabolizing fat within the gut microbiota, producing anti-inflammatory compounds, which we actually confirmed with an analysis of interleukins. We looked at, for instance, we found significance with IL-8, IL-10, IL-1 beta, and I think IL-13. So here we see a lot of communication from the gut into the metabolome. But of course, we see with that PCA, principal component analysis, while we did see separation from those two groups, there was a lot of variability. So this led us to actually investigate one individual who I believe they were over 300 pounds. And they ended up losing 100 pounds over a year. And we had them follow this intermittent fasting diet for 52 weeks. Now, the first 16 weeks were the main weight loss period. I think they hit the 100-pound weight loss at approximately 24 weeks. But from that point, they actually were able to maintain their weight loss. And we see that plotted here with the blue line looking at percent of their baseline body weight. Interestingly, when we looked at the microbiome, we saw it tracking with overall diversity. And when we looked at the overall community dynamics, we actually pull in a lot of ecology with our analyses. So this is Bray-Curtis dissimilarity. So if I were to take, your microbiome sample right now, we kind of consider that your baseline state goes from 0 to 1. So if you were to, take, a heavy course of antibiotics and try to disrupt your microbiome as much as possible, if you completely change your configuration of your microbiome, you would be a 1, right? So what we saw from baseline to 4 weeks is this individual changed their microbiome by over or close to 70%. Now, there was some regression occurring after that initial disruptive phase, but they maintained a reconfigured microbiome close to 50% from their baseline level. And that's really important when we talk about, again, not using the term dysbiosis, but like a disease-associated microbiota. If we want to actually change the overall community in the microbiome to have like health-producing metabolites, what we saw, what we extracted with multi-omic factor analysis in factor 6, that's really important. And there's different therapies available. I mean, everyone knows about probiotics, prebiotics. From our own research, they actually have a pretty small effect, and a lot of the nutrition trials that we run actually have a smaller effect than you think. After you get out of childhood, particularly as an infant, into adulthood, it's actually really hard to change your microbiome. Diet, we've seen with different high fiber interventions, one we actually just published in the Journal of Nutrition, about a 4 to 6 change. And most people go right back because they go back to their old dietary habits. But to see this type of change is really phenomenal. And if we're trying to reconfigure a microbiome, you're going to have to take perhaps some more drastic changes, things like intermittent fasting, which not are only relying on the digestive that's coming in and interacting with the microbiota, but actually having these phases where you're effectively starving the microbiota, which seems to have a lot of beneficial effects, particularly in this group who noted through the gastrointestinal symptom rating scale a significant decrease in most of their symptoms related to gastrointestinal discomfort, which most people in the West actually have. So looking at what was shifting in this individual, we saw some really interesting things. So this is a Sankey plot basically showing at the species level the reconfiguring of the microbiome over time. So this is week 0 baseline. So these are the most abundant species that were in this individual going all the way out to an entire year. So we see obviously a lot of changes occurring, but we also see some prominent features emerge, particularly usually in lower relative abundance, Akkermansia and mucinophilia. And going back to what I was talking about with the mucus layer in your gut and why that's so important, this is one of the microbes recognized really as a steward of the mucus level, because it kind of acts as, like, I guess a lawnmower would be the best way to describe it. It conditions the mucus and in turn produces a lot of anti-inflammatory metabolites that help bolster the epithelial layer and actually also spur goblet cells to start producing mucus, which we want. We don't want an overproduction of mucus, but we want a healthy level of mucus. So seeing these types of changes in this individual was very promising. When we looked at the fecal metabolome, again, going back to that similar plot that I was showing you before, looking at global change, basically, starting from baseline, kind of where they, what their metabolism was configured at, they again saw a pretty stark change, and this persisted over the entire year that they were on this intermittent fasting diet. And we look at a lot of these metabolites, like nicotinic acid, sulfate, different indoles, for instance, which you know have a deep connection to the brain. I'm sure we all heard of the gut-brain axis. We're really showing some positive changes in this individual, and this all coincided with all their normal clinical blood work and, of course, their weight trajectory. So the other side of the coin, of course, we're looking at really the person that started, at the heaviest weight and lost the most. But not everyone is going to respond to these different interventions the same. We performed a responder analysis. So specifically looking at the intermittent fasting group, we are just on this plot to your left, just looking at overall weight change from baseline over the 8 weeks. Now, the blue group didn't do as well as the yellow or the orange colored group, who lost the most weight. So we were interested in what types of differential changes were occurring between these two groups. And what we found, particularly at the functional level, was those that lost the most weight had initially a microbiome that was a lot more attuned to producing anti-inflammatory compounds, metabolizing fat. In fact, a lot of this correlated with particularly visceral fat, which was very interesting and surprising, considering how short this study was. Whereas the low weight loss group really kind of showed a form, I use the term resilience kind of loosely, but you can be in an unhealthy state but still be resilient, meaning it's hard to push you into a healthy state. So while we can look at the really successful cases, what do we do with these individuals that aren't responding as well? And maybe that's not the fairest descriptor, because they still lost, a lot of them, close to 5% of their body weight, which is clinically significant, especially considering the time frame. But this kind of exemplifies a key of what we're trying to do with our particular test. So this is where digital twinning comes in. So the way that randomized controlled trials are going on right now, even with epidemiology, we're just looking at populations, we're looking at groups of people, we're averaging, and we're looking at means, and to an extent variability, but we're not really doing a lot of these large N of 1 type trials. They're complex to do. You have to have a large research team because you're effectively giving people different interventions. This is where in silico bioinformatic pipelines really shine. So has anyone heard of digital twinning? Okay. Can you give us a brief, what you've heard about it?
Well, my understanding is that you take the parameters of individual and just make it on the digital form, and then
Yes.
you can now model and use the different advanced modeling to try to predict, and what you basically transferring the randomized trial from multiple population to n equal 1, and then you do all the study in it, because you cannot experiment on yourself, you will have to duplicate yourself in computer
In
form,
silico.
and then you start doing these experiments on digital form, so you can now pick up the best of intervention. This is what I'm understanding.
Yeah, that's spot on, and that's exactly what it is. Interestingly enough, this technology originated in manufacturing in NASA, because it's very expensive to build these aerospace parts and then do all these types of testing. It was really a cost-saving measure. But as we've had the power of AI onboarded, we can really leverage this, particularly for these really large data sets like I was showing at the very beginning of this talk. So yeah, you're basically mirroring, you have a mirror form of yourself, and this can be your entire, someone's phenotype. It can be everything, or you can look at different segments. You can look at things like the genome. You can look at things like the metabolome. You can even look at the organal level. You can look at, like what we do with our different health module tests, the liver, cardiovascular system, and have all these different domains run digital twins in silico. We're able to do thousands of simulations and compare someone to themselves, seeing, okay, what intervention, what treatment is going to be best for you, what can you expect over a certain time frame, and what you need to do to actually get there. Now, one sampling time point, of course, is a good starting point, but again, to really supercharge these models, the longitudinal sampling is crucial, because that's where we really get good sensitivity as we follow someone's health trajectory over time. So the other exciting feature about a lot of this technology and what it really is amenable to is operating within a blockchain framework, obviously privatized. This isn't something where anyone can go in and start accessing someone's data. A lot of different components in healthcare is trying to move in this direction, particularly with billing, not surprisingly, but also different electronic health records. All that is very intuitive and makes sense. What we are currently working on is actually generating these reports. We have our digital twinning. You can access, in a portal, your health report, but that may not be giving you the information. You may not be getting as granular as you want. So basically, like what we have with large language models, I'm sure you've all used like ChatGPT and stuff like that. So you can interface with basically a chat box and deeply interrogate your report or someone's overall portfolio if you have gained many reports. And we'd like this to communicate with different, not only the metadata that we capture in our survey, but there could also be potential to bring in other health records from that particular patient to bring in and integrate with the data that we're collecting. So say you're running clinical blood panels that you want to see in Congress with the Aristotle or our microbiome test that we're going to have soon. You can even bring in imaging if you're operating in that space. And we even have a translator platform if you're operating in different languages where it can actually translate if that's needed. So this is all very exciting. I'm not going to get too deep into this. We can go over some specifics if you want. But basically what we're showing you here is creating tools that not only you're able to go deep into in terms of the report that you're generating, but also work with the data that you have. So say you want to incorporate the methylation test or the OAT test that you have. I don't know if I would use the OAT test if I have the Aristotle, but you could do that if you were so inclined. So some things in the pipeline that we're working on. Obviously, what I just showed you is something that I'm working on. We're also developing a gut microbiome test, and just like what we termed with the Aristotle, of course, we like to look at luminaries when we name our tests, and we're naming our test the Ilya after Ilya Mekhanov, who was actually a Russian Nobel laureate, and in our opinion, really pioneered a lot of the work in understanding the microbiome. Ilya Mekhanov, he was around in the early 19th century. So, Mechnikov, sorry. Yeah, my Russian's not so good. So.
When do you anticipate having this test?
So I'm not the business guy, I'm just the bioinformatics guy, but we're looking, yeah, we're trying, yeah. Well, that's being charitable. Yeah.
Okay.
December, we're gonna do a pilot with 30 to 50 tests, probably in 3 to 4 months.
Yeah,
Depends on
yeah.
what we can line up logistically, but we'll do an initial pilot with any providers that we have, of about 30 to 50.
So yeah, I'll just give you some, that wasn't the intent of this talk, obviously, but with the microbiome test, we're doing deep genome sequencing, so we're not using like a lot of the common methods that use amplicon sequencing, just again, looking at annotation of the features, so we'll be able to get into functionality. We're combining that with metabolomics of the feces, so we're able to do analysis with what you might be profiling with what's in the blood, as well as several different, I guess less important panels depending on what you're interested in, parasites, inflammatory markers, et cetera. No, so we actually use calprotectin quite often. That's kind of a broad marker. We'll be actually looking at a lot of things associated with interleukins.
Curious question. You just said the word parasite. Sometimes I see patients who are on a bioresonance test and the parasites are common, but when I'm saying go to your doctor and try to find out what parasites you have, because I know you have parasites and you have high SNF.
Yeah.
And there's no test. So will we be able to find out if there is a parasite-specific parasite, or it will be just like, yes.
So there's many different tests that you can do for parasites. So like the older stuff is more like culture-based, or even way back when I guess they crawl at night. So you can do the old tape test, right? But what we're doing is a lot more scientific. We're actually looking at sequences of DNA that we would capture. If they're there, we'll detect them. Yeah, again, it comes down to sample integrity. So we're actually working on a pretty interesting collection method where it's coming from a wipe. I know the microbiome tests have kind of suffered in the past with collection method, and it does have the yuck factor, unfortunately. I know there's been the catch method where you put this little paper catch, and people don't really like that very much. So we're exploring different offerings, but the biggest part is we need to get an accurate sample, which I mean, to be honest, the most accurate sample is if someone sent us their entire bowel movement, which is just not feasible.
Don't ask them.
Just what's
Don't
that?
ask them. They will do something.
Well, some people, yeah. I mean, so this woman, we were at one of the, was it A4M? I can't remember. One of the conferences. I think we went to A4M. Is that where we met you?
No, I wasn't there.
Okay. So this woman comes up to our booth, and before saying her name or anything, she pulls her phone and she shows me a picture of her poop. So I mean, some people are, they have ailments, I guess, or conditions that they'll do anything, right, to solve. Other people, if they're just getting profiling, they're a little adverse to a lot of these different collection methods, which it makes sense. So we're exploring what the best route is for that. Of course, our mobile app, so right now we're generating reports, but this is going to be really important. And of course, an entire portal platform to house the blockchain and really implement what I was just showing you with the conversational aspect of interrogating the reports, and of course, blockchain. So yeah, this was the broad strokes. If you want to go deep in some of the stuff, just feel free to talk to me. I'll give out my card so you can email me all the questions that you have. I think right now, and I always like to put little stupid jokes or little memes and stuff at the very end. In this space, and you're going to hear a lot of these phrases thrown around in the next couple of years. The problem, again, with all of this data is just the sheer amount and understanding, what is actually going to be useful to you. I mean, the degrees of freedom that we now have available with the integration is going to be massive. It's really, to us, the secret sauce is in condensing it and making something usable for you guys.